Yeah, so practical applications of machine learning in biology and drug discovery. So first, we're going to clarify the differences between artificial intelligence, machine learning, and deep learning. And essentially what artificial intelligence is a very broad study of algorithms that are designed to try and mimic some level of human intelligence. So this could include recognizing speech, making decisions, and even understanding natural language. And it's a very broad term. It encompasses machine learning and deep learning. And over here is an example of one of my favorite AI algorithms, which is the rapidly exploring random tree star algorithm. So a lot of times you're left with an arbitrary space and you want to try and find a near optimal solution or an optimal solution. This algorithm is great at that. It's very good at finding near optimal paths. It was used a lot in self-driving cars at some point. I'm not sure if it still is, but very cool algorithm. So next up, we're just going to define what machine learning is. It's basically just a subset of AI that focuses on developing algorithms that can learn from data and make predictions based off of whatever they learned. So it's essentially a way to actually teach machines and making those machines leverage the data they've been trained on. So over here, we got like the family tree of machine learning, and we've got all sorts of different algorithms. Finally, we have deep learning. It's yet another subset of machine learning, and it focuses on developing machine learning models using artificial neural networks. So artificial neural networks were kind of like inspired by the human brain in terms of like structure and function. However, the human brain is definitely a lot more complicated. And essentially the way it works is you have your input nodes and you pass it through these artificial neurons and it propagates all the way through. So this is just a little figure that demonstrates kind of like the different kind of spans of AI. And now finally, let's get into the actual bread and butter of this presentation, which is computational biology. So computational biology is an interdisciplinary field that basically applies computer science and computational related techniques and algorithms to analyze biological data and solve biological problems. So given the sheer complexity and volume of most biological systems, computational biology is practically a necessity when it comes to like modern research. So the insights provided by a lot of different solutions that we're going to be discussing later on in this presentation, they allow us to accelerate a research in drug discovery. They definitely allow us to accelerate research in synthetic biology and all these different fields. And yeah, sorry, let me see what the comment is. Yeah, I'll make the slides available later on. So some general trends that we've been seeing is we've been seeing an increase in number of therapeutics and modalities being developed, especially different types of therapeutics. We've seen a decrease in research time and development time for certain types of therapeutics and certain types of products in biology in general, molecular biology in general. And overall investment in biotech seems to be steadily increasing. It seems to be an overall positive trend, despite all the hype and despite some people being maybe a little bit more bearish on biotech. And then finally, we see a very significant portion of this can be attributed specifically to the adoption of computationally based techniques. So let's talk about some of the applications of computational biology. Probably the most well-known one is protein folding. And probably the main reason why it's so well known is because of AlphaFold. AlphaFold really changed the way people do computational biology. So protein folding models are essentially just models that take amino acid sequences for some protein and try to predict the corresponding structure. The best example is definitely AlphaFold 2. And in this presentation, I'm also going to be lumping in multimer and complex prediction, since AlphaFold 2 can basically do zero-shot multimer prediction. Oh, and over here is a figure of AAUS FP1, which is the brightest known GFP protein. So traditionally, if you were to do protein structure prediction, you'd be using a technique like X-ray or NMR spectroscopy. I'm sure everybody knows this, but NMR and X-ray is very expensive. It doesn't really guarantee results. It can struggle with transmembrane domains or complexes or large samples. There's limitations to these techniques, obviously. And it generally requires specialized equipment and personnel or being able to collaborate with people that have access to those resources. AlphaFold, on the other hand, usually gives you some good insight. It's fairly accurate. It's fast. You can do it within 30 minutes at a very low rate. And it's also highly configurable. And we've been seeing a lot of cases where people are taking AlphaFold or like base AlphaFold and hacking it to do things that it wasn't really intended to do so. In fact, the early multimer prediction, which was done with AlphaFold PTM, this was actually hacked by very astute people on Twitter. So this is certainly very interesting. And obviously, AlphaFold does have limitations, and we're going to be talking about that in a second as well. So just to give a very broad overview of AlphaFold, it basically takes an input sequence as well as an optional MSAN template, And then after this is used to predict an output structure, where AlphaFold will predict all the heavy atoms of that protein structure. So it's quite accurate, especially when provided with a rich MSA. You can technically omit the MSA. You don't have to provide it, but doing so will sacrifice accuracy in most cases. Another thing is one of my favorite things about AlphaFold, and one of the reasons why I think it's arguably one of the coolest architectures in machine learning, period, is the fact that it comes with very good, or well, relatively good confidence metrics that can actually be used to distinguish how trustworthy a prediction is. So some of the limitations of AlphaFold include that it struggles with orphan sequences, so sequences that don't have a lot of homology. You can't really produce a good MSA for them. It struggles with mutated sequences. It's very, very bad at predicting the effects of mutations. And it struggles with like proteins that are very large or complexes that are very large. Although recently, thanks to Multimer v3 that was released by DeepMind, we have seen a very, very strong improvement in terms of like predicting very big complexes and multimers. So this is one of my favorite examples of using AlphaFold because this is an antifreeze protein from the spruce breadworm, and I think it has a very cool structure. So over here we have the PDB experimental structure, and over here we have the AlphaFold version. As you can see, it's actually quite accurate. Another very fascinating case study of AlphaFold is its role in creating a structure of the human nuclear pore complex. This is a very massive complex. It contains over 1,000 molecules, and it's present within virtually all of your cells. One of the main reasons why I think this is very remarkable is because one, it's a huge undertaking, and it was very impressive that people were able to do this. And on top of that, a better understanding of how this giant complex works is going to be critical to developing new potential therapeutics in the future. So now let's talk about some tips and tricks that I've came across when using AlphaFold. So one of the fascinating properties of PLDDT values, by the way, for those of you that don't know, PLDDT is essentially a per residue confidence metric that corresponds to AlphaFold's confidence in that residue's spatial orientation and position in space. So basically, it's like a low PLDT value for a residue means AlphaFold doesn't really know too much about that residue. It might not be like the most accurate prediction. Whereas residues that are, in this diagram blue, which means higher values, they'll usually confer with greater confidence and greater accuracy. So another interesting thing is though, despite PLDT usually correlating with a lack of confidence in a particular part of the protein, it also confers very, very nicely with a lack of order. So inherently disordered proteins or inherently disordered subsections of a protein, they tend to have a low PLDT value. And this is most commonly presented when you look at like the CNN termini of most proteins, because the CNN termini of proteins, they tend to not be conserved. So AlphaFold will usually predict them with very low confidence. And this has some application in enzyme design because some enzymes can be optimized by simply truncating the loose termini. So this is certainly very interesting. Another thing is for membrane-bound proteins like GP1BA. We see that, this is kind of a diagram. It's a protein that plays a role in platelet formation, by the way. And essentially, both of these proteins are membrane-bound, and they have like this long disordered region. And AlphaFold kind of predicts that disordered region very well. And you can kind of use that to almost detect disordered regions just using PLDT value, which is super interesting. Another noteworthy thing to mention is that increasing recycling drastically improves prediction quality in almost all cases that I've seen. By increasing the recycling steps in AlphaFold, you're bound to get a better structure or a better complex prediction. And essentially what recycling is AlphaFold takes its old guess, feeds it right back into the model, and uses that to build off a better prediction. Although in reality, it's a little bit more complicated than that. And another underrated technique is ensembling. So ensembling is very powerful at improving prediction quality. It does increase the actual inference time, but it can also be used to sample alternative conformations in some cases. So it's a cool parameter to play around with sometimes. And finally, AlphaFold 2 can actually be pretty decent at predicting de novo proteins by actually switching over to single sequence mode instead of MSA mode. This was tested by, I believe, Dr. Sergey Ovchikhinov, and this is also where I got this figure. So apparently this also applies to RosettaFold. So this is very interesting. And if you want to learn more about AlphaFold and you want to learn more about different tricks that you can do with AlphaFold, I would highly, highly recommend Dr. Sergey Ovchikhinov's YouTube tutorials on, or sorry, not YouTube tutorials, but YouTube presentations on AlphaFold and Colab design. They're definitely very well put together. Next up, we have ESM-fold. So ESM-fold is a relatively new protein structure prediction model, and essentially the way it works is it uses the ESM2 language model to generate protein embeddings that are basically then fed into a modified open-fold structure module. And this allows it to predict structures with pretty reasonable accuracy, and it's a lot faster because it doesn't depend on an MSA. So while it is faster, it can't predict very large proteins and complexes just yet. But on top of that, it's also what do you call it? It's also slightly less accurate overall than AlphaFold. Oh, and because it uses the OpenFold structure module, it also outputs the same confidence metrics as AlphaFold as well. So yeah, PLDDT, PTM, and PAE. Next up, we have RosettaFold. So RosettaFold was actually an attempt to reproduce AlphaFold by the Baker Lab. It takes more or less the same inputs as AF2, except for it's a little bit faster and a little bit less accurate, kind of like ESM-fold. And another nice thing, another benefit of RosettaFold though over AlphaFold is the reason why it's faster is because it has a lower time complexity from having no triangle attention. And a new version that actually just recently was released, like I believe it was this week or last week or something like that, is RosettaFold 2, which is a little bit more accurate on monomers compared to AlphaFold. So this one is something that I definitely want to play around with a lot more. And who knows, it might have a lot more applications than we would anticipate right now. Oh, and there's also RosettaFold 2 NA, which was released fairly recently as well, and it's a RosettaFold with nucleic acids. Next up, we have inverse folding. So essentially, you're doing the exact opposite of structure prediction. So inverse folding is basically taking some design sequences that fit into a desired backbone. And what that means is usually most inverse folding, models, what they do is they take some protein's backbone, they remove all the side chains, so it's just like the pure backbone of the protein. And sometimes they add a little bit of noise during training time as well as during inference time with protein MPNN. And from there, what you can do is you can predict the actual different kind of like variants of that protein that fit into that backbone. So sequence identity usually varies, but most commonly I've seen it between 0.4 to 0.5% sequence identity. And these models tend to be very fast. They can get hundreds to thousands of predictions within a span of minutes. And it also works very well with complexes. So why do we even want to do inverse folding? Inverse folding seems counterintuitive, but it has a lot of different applied use cases, especially in as an alternative for mutagenesis, so that you can do in silico mutagenesis as well as therapeutic development. So I think this is one of my favorite cases of proteins with very low sequence homology of only 14%, but they have very similar function and very similar structure. In fact, they have the same secondary structures as well, which is 4 beta sheets and only 1 alpha helix. And on top of that, I think it's a little bit better than a lot of traditional mutagenesis techniques, not just because of cost, but because you get a much broader sequence space. With traditional techniques, you usually can only do a couple point mutations. But with mutagenesis, you're kind of making the assumption that proteins with similar structures are going to be carrying out similar function. And this can allow you to search a very broad sequence space. So another interesting application of inverse folding models is actually in development of binders. And we're going to be talking about a little bit more about this at the very end of the presentation. But a good inverse folding model will basically allow you to specify fix and design chains. So which chains you want to actually fix and not touch, as well as which chains you want to completely design. And they'll even allow you usually to specify which residues you want to also fix. And this is pretty effective given like the autoregressive nature of a lot of these models. It's also recently, there's also been applications of these models in development of biosensors and industrial enzymes. So probably the most well-known model in this space is protein MPNN, which is based off the message passing neural network architecture, and it was developed by the Baker Lab. It's very fast, even on CPU, and allows you to specify a fixed chain and all the things we discussed earlier. And most online versions of it also allow options for homomers. So certainly very cool. Another thing about protein MPNN is apparently there is another version that is in development at the Baker Lab called ligand MPNN, which will allow you to also provide ligands into the context. So over here is an interesting example. We have one of our favorite enzymes, beta lactamase from E. coli. And over here is a protein MPNN predicted variant with the AlphaFold 2 structure predicted afterwards. Over here is the native structure from the PDB, and over here is a superimposed structure of both of them. So as we can see, even though you're relying on 2 different models, there's quite a bit of similarity. So it's certainly very impressive. Next up is ESM-IF1, which is the inverse folding model by Facebook. It's quite similar to protein MPNN. It has basically the same strengths and weaknesses, and it's also very fast and allows most of the same features as protein MPNN. It's slightly less accurate than protein MPNN according to sequence recovery, but in my opinion, I don't think that really matters that much because sequence recovery is a somewhat deceptive metric. And we're going to discuss this in a little bit more later on. Finally, there's MIF and MIF-ST. I think this is a great model. It might even be the state of the art for protein inverse folding, especially when factoring in its ability to handle deleterious mutations and potentially harmful mutations in a protein. So out of all the inverse folding models I've tested thus far, it has the best Spearman correlation between the enzyme activity and stability when tested on protein, gym, and some other enzymes. And I'm going to show a figure for that in a bit as well. I believe that much of the improvement from MIF and MIF-ST over the other inverse folding models that we discussed is due to the fact that their training method leverages sequence data in a much more interesting way. So yeah, and another cool thing is this model can basically perform the same downstream tasks as other LMs can. So there's a lot of applications for, there's a lot of potential and applications for using this as a general purpose model. And the other benefit of using an inverse folding model as a general purpose model is the fact that it can leverage structural data, which might be seen as a benefit or a con. So some confidence metrics, both protein MPNN and ESM-IF1 provide metrics in the form of log likelihoods, where values that are closer to 0 tend to be better. And in this case, we generated 100 different variants of the AAUS FP1 protein, and we just plotted the metrics as well as the AlphaFold corresponding metrics as well. We see that protein MPNN is a little bit better at generating sequences, while ESM-IF1 is a little bit better at discriminating between sequences. On top of that, we also see that AlphaFold is very, very bad at discriminating between mutations in a sequence, in protein sequences. So this is the correlation matrix that I was discussing earlier, and it's a Spearman correlation matrix. Essentially, we have how the different inverse folding models correlate with each other. We see that ESM-IF1 and protein MPNN are kind of highly correlated in their outputs when tested on this data set. Also, this data set is, it consists of 12 different proteins. Some are from Protein Gym and some are different data sets from my own team and whatnot. And we found that the best model right now is probably MIF-ST. It's pretty reasonable at discriminating against potentially deleterious mutations. Oh, and these two columns over here just represent like the score metric, the fitness metric that we used. And yeah, MIF-ST performs more or less the best. Protein MPNN performs slightly worse than ESM-IF1, though this really isn't statistically significant. And yeah, that's pretty much the main lesson here. Next up, we have a little plot. This is a figure that I got from the PyFold preprint. So PyFold is another inverse folding model. And essentially what the authors did was they plotted the sequence recovery from the different models and the average inference time or something like that. So what we see is in this figure, it seems that PyFold is probably like the best model because it has high sequence recovery. But when we tested it, it performed probably the worst out of all the models we played around with. And this just goes to show that sequence recovery is a very, very deceptive metric because you don't need high sequence recovery to determine whether or not a protein is actually going to be fit or not. You kind of need a more sophisticated benchmark for this, but sequence recovery is just a little bit deceptive. So I thought I'd throw that in there. Next up is molecular docking. So state-of-the-art molecular docking. Essentially what this is most therapeutics are basically just compounds that ideally have very high affinities for their targets and interact with their targets with very high specificity. So models like DiffDock basically allow you to screen a lot of different candidates, a lot of different potential drug candidates, and at very little cost. Additionally, another interesting application of molecular docking that I'm starting to see a little bit more about is actually using them to design enzymes. So this is very interesting because enzymes are basically just proteins that have a high affinity for some substrate and have to perform an action on that substrate and catalyze it into something else or something like that. So essentially, molecular docking is kind of a similar problem and can be applied. And I think one really cool example was from this paper, which is de novo design of luciferases using deep learning. The authors designed a completely novel luciferase enzyme from scratch. And basically they went into it with like kind of like a desired substrate that they want to do, which was DTZ or anionic DTZ. And in their process, they actually applied molecular docking at some of the stages within their approach. And it seemed to be quite effective, though the docking technique that they used. Yeah, sorry about that, everyone. I actually recently purchased a new microphone, and it seems that this microphone is not very good, so I just switched over to my old one. We shouldn't have any more problems like that, but I'm very sorry about that. It's a little embarrassing. But yeah, so a quick overview on difftalk. Essentially, it's basically just a model that predicts whether or not a ligand will interact with some given receptor, and where it thinks like that conformation is going to be, what it thinks that complex is going to look like. So essentially, you provide it with the ligand usually in format like mol2 or SDF, and you provide it with a receptor in PDB format. And another benefit is it can kind of probabilistically sample alternative conformations by just creating multiple samples of that same complex, which would give us almost like a probability cloud that can be used as kind of a discriminator to determine, how do we think that enzyme is, or sorry, not enzyme, but how we think that receptor is going to be interacting with that ligand. So this is one of my favorite cases of I guess just a ligand bound to a receptor. And this is the crystal structure of LSD bound to the 5-hydroxytryptamine 2B receptor. And over here is the experimentally docked structure in blue compared to the difftalk predicted structure. So one of the reasons why I want to include this example in particular is because LSD is very interesting because its structure fits almost perfectly in a very deep binding pocket of this enzyme. And it's a very interesting drug because you take a very small amount of it, usually in the microgram dosage size, and then after, you're going to be feeling its effect for over 12 to 16 hours. And one of the reasons this happens is because, once the molecule actually gets in, it's very difficult for LSD to actually get into that pocket. But once it does, it's almost stuck there. And there's an additional protein flap that basically seals the molecule shut behind it and kind of folds in on it. So this is very, very interesting. It's one of the more interesting interactions between a ligand and its receptor. Finally, here are like some of the confidence metrics. So difftalk on its own provides some confidence metrics where higher values are correlated with better results. And on Neurosnap, we compute additional metrics using AutoDoc-Vino, which can be used to validate a prediction. So AutoDoc-Vino metrics tend to be inversely correlated with difftalk, with the exception of the intramolecular energy. And stronger correlations tend to result in more accurate predictions, at least from what we've observed so far. Oh, and another thing is over here, this is the same LSD example, but essentially what we did was we asked difftalk to basically make 100 different predictions. We see all of them are pretty much localized around the same pocket. So this could be another metric for I guess confidence, where if you were to see a spread, then we know that maybe we wouldn't actually see that much of a strong interaction. Another thing that I've seen some people do recently, a colleague of mine actually told me about this, is you could try supplying at least an alpha fold. You can't do it with difftalk, but if you can supply multiple ligands, then you can basically do kind of like competitive binding, where if one ligand is predicted to have to go into the desired pocket that experimentally goes there, then you know that it might have a better affinity, at least according to the algorithm in practice. So over here is a correlation matrix of the different metrics outputted in the prior case. And we see that there's a pretty strong inverse correlation between the difftalk confidence, with the exception of the intramolecular energy. So in this case, it seems like it's a very good prediction. Next up is protein language models. So essentially what language models are is they're just language models trained on protein sequences instead of human text and human corpora. So language models include stuff like BERT or GPT and any kind of model that basically learns off of like human text or something like that. And the assumption here is that proteins have their own kind of language that just encoded in a very non-trivial pattern that needs to be extracted and by like some kind of sophisticated model. So in this case, we've seen that this actually works. And as we've seen over the last couple of years, there's a very diverse set of different language models that have popped up, and they all have a lot of like very meaningful representations that they're able to make for downstream applications. So some of the observed applications for these models include predicting protein properties, Go annotations, signal peptides, even predicting stuff like mutation effect and intracellular localization. So it's a very cool and emerging field. And I think one of the language models that might have definitely like played a big role in this field is ESM1B. It's a little bit old, relatively speaking, for machine learning and whatnot. But essentially, it's a general purpose mass language model with around 650 million parameters. And this demonstrated early on that it was capable of learning biochemical properties of proteins. It was even capable of doing remote homology detection similar to that of HH-Blitz. And it's also reasonable at discriminating against potentially deleterious mutations. And it even captures some structural information. So all this was captured, at least to some degree, by just a language model. And this was followed up by ESM MSA 1B. So it's basically the same thing as ESM 1B, except for it now takes it takes an MSA as input. And this basically gives it a lot more context to work with. And we've noticed that it's substantially better at predicting mutational effects. Next up is ESM1V. So ESM1V is just a specialized version of 1B. And it's pretty good at predicting variant effects. And the major difference was that it was trained on UNIREF 90 instead of UNIREF 50. So it allows you to do zero-shot prediction for functional effects of sequence variations. And it's not bad at all, honestly. I've played around with this model, and it's quite good. And it's definitely better when you pair it with ESM MSA. So another thing that that's noteworthy to mention about ESM1V is that it was also demonstrated to improve accuracy when you ensemble it. So essentially, in this particular case, you're training multiple ESM1V models with different seeds, like 5 or 10 or something. And then after you're running some desired input through all 10 of them, and then after you have some technique to kind of sample from the results of those models. And then finally, we have ESM2. So ESM2 is the language model that makes ESM fold. And it's basically just a newer version of ESM1B. It's another general purpose language model. And it's pretty good at all the other tasks. It's also pretty good at predicting mutational effects. And yeah, so the other language model that I want to talk about is CARP 640M. So the main reason why I included CARP is because unlike the other models that we included previously, CARP uses one-dimensional convolution layers instead of being a transformer and using attention like all the other ones. So this makes it substantially faster for training and inference compared to the prior ones, and it's quite competitive in terms of accuracy to the other methods. We've also noticed that it's very, very good for a language model at discriminating between zero-shot for discriminating against potentially harmful mutations in a protein. And another interesting thing is it actually has 10 million parameters less than ESM1B. So it's a pretty great model, and I think there's a lot of potential to actually take this architecture and either scale it or improve it in some other unintuitive way. So if this is something you're interested in, definitely hop into OpenBioML and type in a message or something, because this is looking like a pretty cool project. So here are some other honorable mentions for some language models, their architectures, and the datasets they were trained on. And as we see, a lot of it is mostly just transformers. So here are some bonus stuff. These are just papers that I came across, and I think they're very cool. I think a lot of them have applications, and I think a lot of them are definitely worth mentioning. The first one that I want to talk about is basically creating macrocyclic peptides using AlphaFold. Essentially what this kind of like technique does is it modifies the residue indices of AlphaFold that contain like that encode the positional embeddings in AlphaFold 2. And the way that they do this is with an offset matrix. So using this technique, the authors were actually able to basically predict macrocyclic peptides from scratch that actually form peptide bonds within the N and C termini. So these molecules are fairly small, but the accuracy of the method is pretty good. It's definitely very good. And it's very surprising that they're able to do this with AlphaFold. This is just a perfect example of taking a model like AlphaFold and using it in a very unintuitive way to make it do something cool. And one of the reasons why this is actually important is because cyclic peptides are kind of like an emerging therapeutic modality. They gain a lot of traction. They tend to be very stable, and they have a lot of other very cool properties. So this is certainly quite interesting. Next up is RF diffusion. So I'm sure a lot of us have heard of RF diffusion. It's a very fast model for generating structures given some kind of supplementary information. And it's a diffusion-based model similar to the likes of stable diffusion. So it kind of works in a similar kind of way. A noteworthy thing to mention, though, is that RF diffusion does only predict the backbone, so it doesn't actually predict the individual residues, which is why you have to combine it with an inverse folding model like protein MPNN to actually generate a useful protein. So in this case, this is a diagram that I got from the Baker Labs blog post on RF diffusion, and it seems like they're generating a binder, which I think will be one of the major applications of RF diffusion. So some of the use cases that were discussed within the RF diffusion paper are these, and yeah, it's definitely a very diverse model, and I'm sure there's going to be even more use cases that are going to be popping up after. Next up, I want to talk about at least one artificial intelligence-based algorithm that isn't like machine learning or anything like that. All the other models we've discussed up until now have basically been deep learning models or some kind of variant of a deep learning model. But LightDock and Haddock are pure AI-based algorithms, and they're mostly designed for protein-protein docking. But they also do protein-peptide docking. They do DNA protein docking, RNA docking, and even support membrane-based protein docking. They're both reasonably fast, and they can definitely provide very useful insight. And they're fairly accurate too. Definitely worth checking out. Another thing is that Haddock also supports protein ligand complexes as well as a bunch of other features. There's plenty of features. So definitely very cool algorithm. So yeah, in conclusion, this is arguably one of the fastest moving fields, and keeping up with the latest research in computational biology can be can definitely be a hassle. But at the same time, depending on how you look at it, we're practically living in a biotech golden age. There's tons of room for research, and yeah, there's just always interesting stuff coming out, and there's still plenty of work. So some of the high potential and under researched areas for drug discovery, in my opinion at least, is developing models and algorithms for toxicity prediction. So predicting the potential toxicity of a drug candidate. I think this is a somewhat under researched area, even though there's a lot of work being done in it. I think there's definitely some low hanging fruits. Another interesting one is clinical trial design. So identifying different patient subgroups that are most likely to benefit from some drug, determining the optimal dosage, determining like an optimal treatment duration. All this could fall under clinical trial design. And then finally, we have drug repurposing. So a lot of times you have existing drugs that they might actually have potential applications in other emerging disease areas. So I think models and different solutions for drug repurposing can definitely accelerate drug discovery by just leveraging existing knowledge and resources. Finally, I would like to really give a special thanks to OpenBioML as well as the OpenBioML team. A lot of them, they helped me set up this event. They were patient with me when my microphone broke down a couple of times. And on top of that, they provided excellent insight and excellent feedback for early versions of this presentation. And I also want to talk a little bit about the platform that I developed, which is a zero code option for research and development. It's called NeuroSnap. And essentially, it's a suite of different machine learning tools and pipelines that are now made very easily accessible through a web browser interface, and all the compute is done on our servers. So it's definitely a very effortless way of getting to use AlphaFold or DiffDock or any of these other models. It's if you want to do something remotely, it's definitely one of the best options as well. And yeah, I hope you guys all like this presentation. I recently made a LinkedIn, so you're all more than welcome to connect with me. And on top of that, I have a blog, so I've been posting some articles related to machine learning and biology and whatnot. And yeah, I really hope you guys enjoyed the presentation. Thank you.